My name is Pauli. I'm a postdoctoral fellow at Cornell University. I work with Michael Gore, and we work on high-throughput phenotyping. And what I'll be talking about today is the work in cotton. I realize it's not wheat, but hopefully an hour-long break from it won't kill you guys. Just so I do have some street cred, I got my PhD working on malting barley. I don't know if that counts for anything in the wheat crowd, but so briefly today, I just want to give you guys a brief cotton background because most of you probably aren't familiar with the crop, and it is quite different than the traditional cereal crops. And then talk about the high-throughput plant phenotyping experiment, the platform, and then kind of just some practical aspects of processing the data and using it for the actual QTL mapping, and then kind of the thoughts and, I guess, about future directions with regards to plant improvement and integrating all these various data sources. So in terms of cotton production, the top three are China, India, United States. China and India are battling quite ferociously for who's number one. It changes from year to year, but overall, cotton is produced in over 100 countries. It's the number one fiber for obvious reasons. I'm pretty sure everybody has a piece of it on them right now. And so there's two main species. There's upland cotton, G. hirsutum, that's 95% of the world acreage. And then there's also Pima cotton, which is also called Egyptian cotton or sea island cotton. But what's really unique about this species is that, it's a perennial tree. And so this is what makes it different than like small grains, is that management is huge. It's just about as big as environment in terms of keeping the stature short, getting it to flower, the fact that the bloom lasts a long time, and then finally applying defoliants. The fiber itself is a single cell, which makes it kind of unique in the sense that it's all about depositing cellulose, and then at the end of the season, after this deposition has gone down, it shrinks, it dries, it twists up, and that's what gives you actual fiber strength. But the fact that it's pure cellulose makes it nice in the context of doing the actual physiology about converting carbon into pure starch. So with most things, sustainable agricultural production is threatened by variable weather patterns as well as diminishing freshwater resources. This is a farmer in Texas in 2011. And here's a map of the cotton production in the U.S. overlaid with the drought index for that year. It was one of the worst droughts ever, $2.2 billion in economic loss. 55% of the acreage was lost as well. And I apologize for making this a state-centric slide, but the global data supports this in pretty much all production regions for cotton. So now on to the actual high-throughput phenotyping experiment. Here is the current platform that's being used in Arizona. You'll see at the front there's the array of sensors, and then there's a cab. And the thing that's unique about this setup is not only do those sensors rise and lower, but the tractor itself is actually on hydraulic lifts. So it is very conformable to the conditions of the crop in case the plants get rather high. It has been revamped. It now sports its own air conditioning unit, not for the driver, but it actually sends AC air out to the sensors themselves to maintain sensor body temperature. And now it also has an onboard, essentially, supercomputer to process the data real time, as well as run some of the instrumentation. But back in the good old days, this was what was used. Not nearly as fancy, but what was on board was an infrared thermometer to measure canopy temperature. We had an ultrasonic transducer. This measures plant height. And finally, a crop canopy sensor. The crop circle is what we chose, and that measures your vegetative indices. But one of the key components about this system is the ability to geolocate every single data point to a spot in the field. Your traditional GPS will not be able to do this. The standard resolution is about 3 feet to a meter, which just doesn't work when you consider that these rows are probably less than a half a meter apart. So there's a thing called RTK, which stands for real-time kinetics. And basically, this takes your average GPS and puts it on steroids to the point where now the precision is down to the centimeter level. And so that's really what's key in terms of making sure that the data points line up with what's in the field. If not, all this work, all this data collected is pretty meaningless. Also, we have the onboard data loggers, and the tractor can go through the field pretty quick, collecting about 1 data point per meter. You can obviously turn up the sampling frequency to achieve more. In our case, we could do 440 plots in about 30 minutes. And so here's the actual experimental design that was used. The experiment was conducted in central Arizona, which is an optimal environment. Matthew alluded to it Monday about problems with canopy temperature in terms of ambient humidity as well as cloud cover. And so in Arizona, you have pretty much none of these factors. It's continuous clear skies, limited rain, high temperature, so it provides a very consistent stress environment for doing this type of work. It was a rather small population, only 95 lines. And we used two treatments, pretty much a well-watered and a water-limited. And how this was done was using crop modeling in terms of the actual irrigation applied. We had neutron probes buried in the field to collect the data and make the appropriate adjustments. And the stress treatment was applied at and after flowering to minimize the effects of phenology. In terms of the data collected, we had canopy temperature, reflectance, height, and physiological traits included abscisic acid content, carbon isotope discrimination, a whole bunch. There's probably over 50 phenotypes for this data set with another 10 coming. And so here's actually just an image of some of the geoprocessed canopy temperature data. Each one of those little circles represents a data point. And so the green are the warmer temperatures, blue is obviously cooler. But one of the first thing I guess that struck me is you notice that with this type of setup and collecting the data, you can see the within field variation specifically down here. And this area of the field actually had a high clay content, which holds a lot of water. So that was one of the advantages of having the system is the data you collect actually informs you about the environment that the plants are experiencing. I just wanted to give a quick note about how we're using NDVI in this experiment. We're not trying to quantify the greenness, if you will. We're using it to quantify the stress response of wilting. Basically, when plants have enough water, they're at full turgidity, and the leaves are perpendicular to incoming light. But as the stress is applied, they lose their water, they become wilted, and you can see in the photo, they start to hang down. And because of this, there's more infrared light coming from the soil. And that response is what we are trying to capture. And so basically the next step is taking all this data that was collected on the platform, putting it through the analysis pipeline in order to get, identify QTL that can be useful. So one of the things that has to happen is we have to do the geoprocessing of all these data points. And basically make sure that they map up to the right spot in terms of where they are in the field. So the blue boxes are the actual plots. The little red dots are the data points for canopy temperature. But you'll notice there's this circle in the middle. Does anybody have any ideas what that might be? No, it's a circle, right? It is. This is just an artifact from the tractor stopping in the field. And anyway, these kind of issues arise, and they're kind of hard to catch in a traditional modeling sense. So it's having somebody actually sit down and kind of go through these data sets and look at the quality and make sure everything looks good. In terms of actual data preparation, as always, it's the most important step, but it's also the longest and least fun. With canopy temperature, this wasn't alluded to, but one of the biggest things is the effect of the ambient environment as the sun is moving across the sky. And you can see in the graph that just in that 30-minute window, the ambient temperature is increasing about 4 degrees. And if you do not correct for this, there's pretty much nothing you can really get from this data. And so kind of the issue is collecting high-throughput phenotyping data, one of the key things to have is a timestamp. Autocorrelation is a real thing. It exists, and especially when you have this context of collecting that much data in a given time. The sensor performance changes as a function of temperature, humidity, and so being able to account for that is critical. And so you can do it in like a traditional ANCOVA method, or use time as a random factor. It's kind of what's done in econometrics, and it worked quite well for us. And then obviously just plotting data to catch any issues that arise. Like in this instance, our plants were taller earlier in the season than later. So obviously something was wrong there. And it's, if you don't catch this stuff, the QTL analysis obviously is going to be flawed. And then so in terms of outlier removal, so you've done all this and you still have what you think is a good data set. And then you still have to go through and get rid of outliers that are having an influence on your actual model fitting process. And so how we achieve this is we use a mixed model approach. And for this, we actually use SAS, which was not the best choice. It's rather slow, a little finicky. But you can see that after removing those outliers, things change drastically. On the right-hand QQ plot, you'll see that there's still some bowing on the ends of the distributions. This is mainly an artifact from mixing the wet and dry treatments together in one plot. Overall, the data came out quite nice in terms of being normally distributed and usable. And finally, I would just make one more statement about the field variability. For us, we use subsurface drip irrigation. And so this gave us, pretty discernible irrigation flow direction. And so making sure you account for this is pretty critical. And so I guess, it was all experiments in plant breeding. Having an adequate and experimental design that you, feel confident in being able to correct for any problems is very important. Whatever model you want to use in terms of the spatial model, moving average, autoregressive moving average, just as long as you have a handle on what is going on in the field, it's pretty critical. So next, the actual genetic analysis of this data. And mostly I'll be talking about canopy temperature. It was alluded to on Monday. It's a very indicative trait of what's going on with the overall health of the plant. We did have the reflectance data. It turned out quite well too. But for the purposes of this talk, I'll just stick with canopy temperature. And so this is just the plots of the distribution of temperatures that we observed in the field. This was at 1 p.m., so this would be at pretty much peak stress. This is when vapor pressure deficit is going to be the highest in our environment. And anyway, you'll see that we get a pretty clear distinction between the two treatments, about a 10-degree difference. The standard deviations are approximately the same, a little bit wider in the water limited. Basically, the stress being able to suss out the differences between the genotypic responses. And I do want to just clarify that the water limited plots, it's not like they're out there dying because they have no water. They're still being irrigated, just obviously at a reduced amount. You might notice, though, the heritability estimates for this trait are rather high, 0.83 and 0.9. And obviously, this is an artifact of using a small population, but really what's driving it is the fact that we can sample multiple times in a day, and with the experimental design, we're getting 8 observations per line. And so going back to the heritability equations, high replication is always your friend, and that's kind of what's driving this. And also the fact that we're using a multivariate model, and so we can actually include an air variant structure, further extracting some residual variance. And that's kind of what's driving the high estimates. But the main take-home is that these traits do have high heritability, and there is genetic variation, which is critical for plant improvement, or else you can make no genetic gain. And looking at the differential time by treatment interactions, we see on the bottom canopy height, pretty much as we expect, if not very responsive. You just see the differences between the two treatments. We calculated leaf area index using a compound canopy index, which relies on pretty much reflectance values, NDVI, and canopy height. And you can see that in the water-limited treatment, it kind of goes down over time as you would expect. But it's more pronounced with NDVI. Pretty much by 10 a.m., you're already seeing the effects of water deficit. And then by 1 and 3 p.m., they're well established. You might notice that the well water treatment, the NDVI values actually increase over the course of the day. And this is due to diheliotropism. It's a function of cotton where the actual leaves track the movement of the sun in the sky. And then finally, canopy temperature, pretty much what we expect. At about 1 p.m., we see about that 10 centigrade difference. It's pretty substantial. And so this kind of just gives us a whole idea of the experience that the plant is having in terms of the stress that it's under. And because what's neat is that cotton, I don't know if I can say this categorically, but remobilizes other parts of it, like the root structure is essentially cannibalized and moved up to the canopy. And as it goes through these stages, in fact, during bulb development and fill, the photosynthetic apparatus is being reduced and the leaves are being remobilized, the nutrients, into bulb development and fill. And so what's also unique is that this flowering period, in wheat you're used to just, this set time window. And with cotton, this window, it can be much longer, including the entire season, depending on how the crop is managed. But there are still very distinct growth stages in cotton in terms of flowering, boll development, and then fiber development, which occurs at kind of the end of the season. And this is when most amount of the carbon is being fixed. A typical plant per day is fixing roughly 7 grams of CO2. And so here's just kind of a plot illustrating the dynamic response of canopy temperature to the irrigation regimes at multiple days and times throughout the season. And pretty much what we see is we get this continuous response. It's very well defined, and regardless of the growth stage of the plant's in. And what's neat about this is that when you do the correlation analysis, the readings taken within a growth stage are more correlated with themselves than across. So this gives us an idea that there is some underlying biological process that's occurring in the canopy that the thermal signatures have that we're picking up. But more importantly, it also really highlights why continuous phenotyping throughout the season is really needed to capture what's going on with the plant. That just static time points aren't going to be sufficient. And so we go through the actual QTL analysis, and we see these distinct temporal patterns. And so I realize this graph is probably a little bit hard to interpret, so let me walk you through it. On the top is the water limited, the dry treatment. On the bottom is we call it wet, the well watered. On the x-axis are the different chromosomes, with the genetic positions of where the markers are at. On the right-hand side are the years in which the data was collected, and on the left-hand side are the days within the years. And so if it's a red colored, it means that it increases canopy temperature. If it's blue, it decreases it. And the color intensity, it basically reflects the LOD score of the QTL detection. And so just two I'd like to highlight are on D4 and D12. We got these very distinct QTL patterns. And these QTL would show up not during flowering, but pretty much only boll development and fill. And then the expression would go away in some cases. And so this was kind of encouraging because it really does highlight the fact that our traditional approach of taking single time point measurements and static data does not reveal the entire story of what's going on. I mean, if we would have collected this data during flowering, we would have missed this. And so what this really highlights is that with high-throughput phenotyping and collecting the data and having basically a longitudinal assessment, we can kind of see the trait development as opposed just to the cumulative effects of QTL. And so that kind of gives us an idea of what to optimize in certain environments. And so with all this high-throughput phenotyping and all the work that we're doing now, and I think as a community, the important thing to remember is the data we're collecting have real biological significance? Like, is the signal we're detecting real, or is it just like an artifact? If it means nothing, then we probably shouldn't pursue it. And so luckily, the cotton genome came out about 2 weeks after I finished this. And so we were able to go through and take our QTL intervals and query the genome to see what we could find in these intervals. And for carbon isotope discrimination, which I'm showing here, we found this drought-responsive family protein basically was implicated in stomatal aperture and regulating that. The other trait that we collected was abscisic acid content. And so, this canopy temperature QTL mapped to the same region as this ABA binding protein. And so this was extremely encouraging because it means, yes, what we're measuring is an actual effect. It's a product of, the physiological response that the plant is generating. In addition to these ones, we had a heat shock protein. We had a QTL for lint yield that mapped to canopy temperature. That was pretty much a lipoprotein involved in membrane stability. And so I guess, this is the important part, making sure that the traits that we're measuring with these high-capacity systems are actually capturing biology. And so Monday there was some discussion about transpiration, whether you want high, low, depending on the environment. And for us, in this case, cotton in Southwest America has been bred to transpire quite heavily because it's generally always irrigated because the conditions are so extreme. And so in this case, the high leaf transpiration does provide an adaptive advantage, provided that there's available soil water. And so this kind of brings up the idea of, do you breed for water use efficiency, or as Abraham Blum says, efficient use of water? I guess, that's the distinction for the plant breeder to make. But moving forward, we see that we have this response and that we're able to detect it. Now, finally, a canopy temperature, the correlation with some of the agronomic traits. Canopy temperature at flowering and peak bloom was most predictive of lint yield, which is not surprising given that if you don't have a flower, you can't develop a boll. So this was pretty, it's cool in the sense that for other traits like boll size, another driver of yield, it was most correlated during boll development and fill with canopy temperature. So combining these different indices gives us a predictive way of hopefully understanding yield development in cotton. And so one of the other sensors that we've incorporated is a LiDAR. It stands for light ranging and detection, and basically it's a sensor that sends out a photon and measures the time it takes to bounce back to the sensor. And so here's a scanning image of pretty much a cotton block. You can see the different rows divided up into ranges. When we zoom in, here's the actual cotton plants. And I would just like to say that these are defoliated plants because the light can't penetrate leaves. But we're beginning to see the actual structure of the plants. And when we go even further, we get like a very clear pattern in terms of boll distribution, structure of the plant. Because cotton is a tree, there is genetic variation in terms of the actual structure of the plant, whether they call it a stovepipe versus diamond, and basically how these are distributed on the plant. And more importantly, this kind of gives us a new phenotype with which to work in terms of mapping the distribution, the number, because for different environments, you can hopefully manipulate where you want your cotton bolls on the plant. For some environments like the high plains of Texas, they have extreme conditions, and they only will get 2 or 3 cotton bolls per plant if they're lucky. And depending on the type of machinery they use, it'd be nice if the bolls were higher up on the plant instead of in the dirt. High intensity areas like Arizona, not so much of an issue. And so just kind of in summary of this, development and deployment of the sensor platform, it took quite a while, but the data that we generated from it was pretty good, and kind of gave us a very unique insight by being able to collect it numerous times of the day throughout the season. I think the most important thing for us, though, was it gave us a glimpse into trait development as well as phenotype development, as opposed to just these static time point measurements that really don't reveal the dynamicism of what's happening with the phenotype itself. And I guess finally with connecting that genetics to physiology, I think that is something that as we move forward as a community to always keep in mind that, our data is actually capturing something real. And so in terms of moving forward, this is kind of what our group is working on and some of the directions we're taking. And I just thought I would share this with you just to show you what's possible, what's available. So here's an image of northern leaf blight. And has a very distinct lesion pattern. It doesn't show up. It's pretty much these brown ones on the top side of the leaf. And this is a serious disease in high production areas, especially in the Northeast, where we have high moisture. And so now you guys have looked at that, and you've burned this into your mind. Of which one of these 4 photos do you think is exhibiting northern leaf blight? That's a challenge. And so kind of where we're moving is image data hasn't really arrived yet, but it's on the way. And I'm speaking strictly from the field sense. The high-throughput phenotyping cabinet systems that are available, they can do all sorts of stuff. But for us in applied breeding, it's basically what you can measure in the field. And so this highlights the challenge of collecting image data in the actual production environment as opposed to controlled setting. And so this is kind of where we're going now and what we're working on. And just to clarify, it's this one on the top left was the one exhibiting the true northern leaf blight, right? And so, as image data comes online, and we have more and more of these sensors, what we're finally going to be moving to is actually mining video. I mean, that's kind of the next thing that's coming up. These, images and video represent probably the richest data stream there is in terms of capturing information. And if you're, able to do this on a continuous basis throughout the season, I feel confident, we will be able to learn a lot of things. And so one of the questions is we're going to collect the data, but how are we going to use it? And for us, it's the application of machine learning. I realize this is kind of one of the buzzwords going on right now. Apologize for that, but it's what we're using. It works. And so what we're doing is we're going to collect a bunch of data and let the math do the work for us in terms of extracting new phenotypes. The human eye is good at recognizing patterns, but when you are trying maybe to detect things that are minor, maybe predicative of what's going to happen to the plant, like stress physiology, like in maize, you can see very distinct vein chlorosis sometimes when plants are stressed. And so the idea would be, if you can extract that feature and develop some type of predictive model of that. And basically, machine learning opens up this avenue of detecting latent variables that maybe we're not even aware of that are important. And so if we can implement this, we can integrate it directly into BLUP estimation, genomic selection, estimated breeding values. And moving forward for us, this is a project that we just recently started, and it's pretty much incorporating artificial intelligence combined with drones. And what we'll be doing is having a drone that flies the field. In real time, it's collecting image data. It's sending it back to a central processor. It's running this algorithm, and it'll tell us whether or not it sees northern leaf blight in the field, and if it does, how much is there. And working on disease traits is probably the easiest one for us first. But I think it'll be quite valuable because I think we were joking the other night about some of the Rust scales, about how there's plus signs and semicolons, and what does that mean? And so, using this type of approach gives us a way to actually quantify disease resistance. And some of the early demo data that we worked with was stem rust. And it did an excellent job of, saying this is stem rust, this isn't. And so that's kind of where we're moving forward. And, integrating, autonomous robots, working with humans. I know in the picture, it doesn't look so good because the robot, the drones are kind of our overseers, and you got the guy out in the field shoveling. Yeah. But I think it'll be a very useful tool. And so, that's cool, but in terms of actually having an impact, I think most of us would agree that one of the reasons I think most of us get into plant breeding is you want to have an impact, you want to help people. And so this stuff with the drones and the cameras, it all works really great as long as you have a big computer and all the resources, but most people that really need this technology don't. But what they do have is a cell phone. And so Mike and I and some other people in our lab are collaborating with folks in Uganda in terms of developing these, basically smartphone apps that take an image of, in this case, a cassava leaf, and the algorithm runs and says, hey, you either have cassava mosaic virus or you don't. And for the people that really need this, that's huge. That's something, if you can catch it early and change your management strategy, that's a real boon to those people that need it. And I don't know, it is cool to think about the stuff that we can do, but if it's not in the context of really helping those that truly need it, then maybe it's not always worth it. So this is kind of the direction we're going with using machine learning and images. One of the other things we're working on is, physiology in G by E. And so one of the projects we have is seed ionomics of cotton. And basically, how does the plant extract elements from its environment, and how does it incorporate them? Monday, Matthew made the statement that phenotyping roots in the field is probably not worth it. I hope I don't, paraphrase him wrong, but and in some sense, I would agree. At this current moment in time, the technology in terms of what's available to do that is is limited. The ground penetrating radar, I've seen the same images, and it looks like it would work well for potato. But for most crops, no, I don't think it's a viable solution yet. But so if we don't need to phenotype the roots themselves, but I think what is critical is to understand the environment that those roots are in. Most of the time, all of our phenotyping work for the past essentially 60, 70 years is focused on what's above the ground, when really what's below is equally, if not more important. And so we're moving in that direction in terms of figuring out how plants change their physiology and how they interact with their environment to cope with the stresses that they're enduring. And so one of these things is using ionomics. Obviously calcium and potassium are two of the biggest signaling elements in plants. Calcium driving stomatal regulation. So understanding how these are incorporated in the plant is pretty pivotal. And at this point, we're beginning the preliminary data, and this biplot of the actual traits. The big thing just to notice is that things change in response to the environment in terms of the water that they're receiving. I won't pick out any specific elements that are really cool, but calcium is eliciting effect, as well as potassium, most likely because these are involved in osmoregulation and monitoring the plant-water balance. And so the next area that we move into is this idea of envirotyping. I think it was coined by Mark Cooper. I've been told that, but I'm not sure. And this is kind of, if we want to capture, if we want to understand G by E, we need to know what the E is. And again, atmospheric data, I'll just skip ahead one, but we've been pretty good at that. I don't know how many people have these watchdog weather stations. They're awesome. It's about $1,500. You stick them out in your field, collect data nonstop for the whole season. But $1,500 is still quite a bit of money when compared to, on the right-hand side, you have this low-cost solution. It's $165 to build one of these. It's driven by an Arduino, or you can get a Raspberry Pi. It's measuring soil temperature, soil moisture, relative humidity, air temperature. You can do canopy temperature. And so pretty much you can build numerous of these and just put them out in your field and really actually characterize the environment that the plants are experiencing. Going back one with the soil, on the left-hand side, this is a EM38 image, and basically EM38s are a sensor that you drag across your field, and it measures conductivity. And this gives you an idea of basically the water holding capacity of the field in terms of clay content and how much water will be there for the plant as the season progresses. On the right-hand side, so in addition to collecting ionomics data on the seeds themselves, we actually collected soil samples at 5 different depths and did the ionomics analysis on those as well. And so in this image, obviously we didn't sample every plot for the soil. This is just a Krieged image of interpolating basically what those levels, what those values of calcium would be throughout the field. And so this kind of gives us the idea of the field variability, and this is critical when we actually try to go and model the response. Because if you just work with the seed ionomics data, you have no idea about what access it had to calcium. Like maybe it was in this part of the field where it had a high amount compared to somewhere it was low. I mean, that was driving your response. So getting a handle on the environment that the plants are experiencing is critical. And then so kind of tying it all together is the applications to plant breeding. And it's kind of the intersection of genotypes, phenotypes, and envirotypes. And I changed my figure because Graham showed this one from his paper on Monday. I was using the soy sim model that's been developed. And so I like his image because at the top, the second layer, it has like the parameters. And what crop models really represent is a way, it's a functional relationship between the plant physiology and the environment and how that leads to formation of yield. And I think this is where high-throughput phenotyping comes in, because what we really have to do is estimate these growth parameters, these inputs that we're giving the model. And so in terms of like leaf appearance rate, using LAI, we can actually get genotypic values instead of just Because traditionally right now, you go out and grow like maybe 5 varieties and phenotype them intensely. That doesn't really always give us a true story, but if we can develop, growth parameters for each genotype, it becomes a very attractive option. I mean, plant development is dynamic, phenotype development is dynamic, and so our data sets need to be dynamic, or else we're just not going to see the whole picture. And so moving forward, if we can use the crop models and we can use high-throughput phenotyping data to extract these growth parameters, A, they become new phenotypes, but B, they also allow us the potential to establish these growth curves for each genotype. And you'll see the 3 lines. I mean, plants achieve their final yield by different methods. Some flower early, late, depending on stress or the environment. And so what's driving that, obviously, is the different underlying genetics. And if that's what we want to capture, it only makes sense that we need to be able to monitor that, continuously. Because like with the cotton stuff, we know that there's QTL that aren't expressed all the time. And undoubtedly, that's the same situation in most crop plants. And so, if we can do QTL mapping on a continuous scale, we can also incorporate crop growth models into genomic selection. Again, Graham mentioned this paper Monday, but unfortunately he ran out of time, so he didn't really talk about it a lot. It just came out in June, and what these guys have done is actually integrated the crop growth model into their actual GEBV estimation. And what they're able to do is take, the genotypes, add environmental information, and predict into new environments that they had never even tested. And their accuracies are shockingly high, I would say. And so as we move forward, I think this is kind of the context that we are operating in. So the same thing where on the bottom, we have QTL expression in terms of length, magnitude, and we have that in relation to the environment about when precipitation is occurring, when peak temperature stress is occurring. And so by doing this, we can come up with these genomic estimated breeding value curves, allowing us to optimize genotypes not only for environments, but essentially within season variation. Basically taking a target region and making it smaller. Because climate change is a real thing. It's making, variable crop production challenging. And, majority of us aren't producers. And we don't have to face the reality that growing, farming is extremely risky. It's challenging. And anything I think that we can do to minimize those risks for the producers is worth doing. And then finally, as we move forward, there's this idea of autonomous vehicles. Currently, I realize that UAVs are very, hot. Everybody's got one. They show cool images. We've all got our own, I guess. But I would say they're just a temporary thing, kind of. Autonomous vehicles offer us a lot of things. Basically, continuous phenotyping throughout the season, even at night. For us in the States, we don't have to deal with the regulatory hassles of flying drones, which is just extremely burdensome. It's like a 9-month period to get the regulations approved. In this case, we can actually get physical sampling of plants. This image, this is called the Bonnie Rob. It was developed out of a group in Germany. Fully autonomous, can actually go down, take a leaf punch, collect data for you. It works well with corn. And then more recently, we've kind of started this project with the folks at the University of Illinois for sorghum. And we have an autonomous vehicle that will go through the field and images the plant at numerous heights and actually creates a 3D reconstruction of the entire plant. And so, that actually provides you, limitless phenotypic data. You can extract leaf angle, width, height, volume of the plant, and that's really what we want in terms of figuring out these growth parameters and hopefully, moving into the context of incorporating these plant growth models. And so again, just, a richer data source. And so I think all these things combined, we're at a very pivotal time in terms of, high-throughput phenotyping will undoubtedly impact the way we breed plants, but so will all these technologies that we're talking about. It allows us to look at, plant development and expression in a new way, kind of on a longitudinal basis, as opposed to static time points. Again, for us, it's mainly getting to see trait development as opposed just to trait accumulation, and kind of a new idea in terms of dissecting the genetics of this. Finally, integration of all these with the use of crop models. I think, Identifying growth parameters using those as phenotypes will be very valuable. And getting a handle on G by E. We're very good at predicting additive effects and working with additive models and things that are linear. But the reality is most systems aren't, including plants. So this kind of gives us a good way to achieve that. And then finally, breeding for precision environments in terms of optimizing genotypes for the environment that they're going to experience, even down to maybe the time scale in which the stress they're going to experience occurs at. And finally, variety optimization and bringing benefits to producers and consumers. With that, undoubtedly, there's always a lot of people to thank. This is just but a short list. And with that, I'd like to thank you for your time, and if you have any questions, I appreciate it.